In this video, you will learn how to define and contrast mitosis and meiosis. The reproductive system produces the next generation of offspring. Reproduction requires specialized cells called gametes, the sperm and egg cells. Sperm cells are also called spermatozoa. Egg cells are also called ova. While spermatozoa are the male gametes, the female gametes are ova. Gametes are haploid cells, meaning they contain one copy of each chromosome. In humans, haploid cells contain 23 chromosomes. When fertilization occurs, two gametes combine. 23 chromosomes come from each gamete. Together they form a new cell called a zygote. The zygote is a single-celled embryo. Unlike gametes, the zygote is diploid, meaning it contains two copies of each chromosome. In humans, diploid cells contain 23 homologous pairs of chromosomes. A pair of homologous chromosomes contain the same genes. However, they may have different versions of these genes, known as alleles. After fertilization, the zygote begins dividing through mitosis. Mitosis produces most of the cells in the body. These diploid cells are called somatic cells in contrast to the haploid gametes. Each mitotic division produces two daughter cells with identical alleles. In contrast, meiosis involves 2 rounds of cell division producing 4 genetically unique haploid gametes. Most cells of the human body are diploid somatic cells that contain 23 pairs of homologous chromosomes. Males have one X chromosome and one Y chromosome. During DNA replication, each chromosome is copied. After replication, each chromosome has 2 identical copies. These copies are called sister chromatids. Sister chromatids remain attached at the centromere. Now we are looking at replicated chromosomes from a female. Females have 2 X chromosomes. All 46 chromosomes have been copied by DNA replication, so each chromosome contains a pair of sister chromatids. Before a cell divides, it progresses through the cell cycle. The cycle begins with a newly formed cell in the first growth phase. During G1, the cell grows and performs normal functions. If a cell is not actively dividing, it can enter G0. Some mature cells remain in G0 and never divide. If the cell is going to divide, it will need to copy the DNA. This occurs during the S phase. S stands for synthesis because DNA replication occurs during this phase. Next, the cell enters the second growth phase. During G2, the cell checks for DNA damage and produces proteins that prepare for division. After G2, the cell enters the M phase, which includes mitosis and cytokinesis. Mitosis divides the nucleus, and cytokinesis divides the cytoplasm. Mitosis occurs in 4 phases. These phases are prophase, metaphase, anaphase, and telophase. You can remember these stages by following what happens to the chromosomes. First is prophase. During prophase, chromatin condenses into visible chromosomes. At the same time, the mitotic spindle begins forming. Then the nuclear envelope breaks down and spindle microtubules attach to chromosomes. Next is metaphase. During metaphase, chromosomes align at the center of the cell. This center line is called the metaphase plate. Then during anaphase, sister chromatids separate. The mitotic spindle pulls them toward opposite sides of the cell. Finally, the cell enters telophase. During telophase, nuclear envelopes reform and chromosomes decondense. After mitosis, cytokinesis divides the cytoplasm and 2 daughter cells are produced. Now let's contrast mitosis with meiosis. Here we see specific examples of cells dividing by mitosis or meiosis. The spermatogonium divides by mitosis, producing two genetically identical cells. One of these cells differentiates into a primary spermatocyte as it enters meiosis. Meiosis produces four genetically unique gametes. Unlike mitosis, meiosis includes two rounds of cell division. These divisions are called meiosis I and meiosis II. In meiosis I, a primary spermatocyte divides producing two secondary spermatocytes. Secondary spermatocytes are haploid cells, but they still contain pairs of sister chromatids. In meiosis II, each secondary spermatocyte divides producing immature sperm cells called spermatids. During prophase I, homologous chromosomes pair together and crossing over occurs. During crossing over, homologous chromosomes exchange DNA segments. Then spindle fibers attach to chromosomes. During metaphase I, homologous chromosome pairs align at the midline. Next, during anaphase I, homologous chromosomes separate. Each chromosome still contains two sister chromatids. During telophase I, two nuclei form. Each nucleus contains 23 replicated chromosomes. Cytokinesis then produces two daughter cells. Now meiosis II begins. Meiosis II closely resembles mitosis. During prophase II, the spindle forms again. Then in metaphase II, replicated chromosomes align at the midline. During anaphase II, sister chromatids separate. Finally, in telophase II, the nuclei form and cytokinesis divides the cytoplasm, producing four genetically unique cells. So remember the most important distinction. Meiosis I separates homologous chromosomes. Meiosis II separates sister chromatids. Meiosis produces genetically unique cells. Two major mechanisms create this genetic variation. The first mechanism is crossing over. Crossing over occurs during prophase I. During crossing over, homologous chromosomes exchange DNA segments. This process mixes maternal and paternal genetic information. The result is new combinations of alleles. The second mechanism that creates genetic variation is independent assortment. Independent assortment occurs during metaphase I. Homologous chromosome pairs align randomly at the midline of the cell. This random orientation determines which chromosomes enter each gamete. So remember, crossing over shuffles genes within chromosomes, and independent assortment randomly distributes chromosomes into the gametes. The production of gametes is called gameteogenesis. In males, this process is called spermatogenesis. Spermatogenesis produces sperm cells, also known as spermatozoa. In females, gameteogenesis is called oogenesis. This process produces ova. Both processes begin with mitosis. Mitosis produces diploid stem cells. In males, these cells become primary spermatocytes. Primary spermatocytes undergo meiosis, and each primary spermatocyte produces four haploid spermatids. These spermatids mature into spermatozoa. In females, mitosis produces primary oocytes. Primary oocytes enter meiosis during fetal development before birth. However, meiosis I arrests in prophase I before birth and doesn't resume until after puberty. Then meiosis II arrests before ovulation and doesn't resume until after sperm penetration during fertilization. In contrast to the four spermatids produced by spermatogenesis, only one functional ovum is produced by oogenesis. The other cells become polar bodies. Polar bodies remove excess chromosomes and eventually degenerate. So remember this contrast. 4 sperm are produced from each primary spermatocyte. Oogenesis only produces 1 ovum. Now let's summarize the key differences between mitosis and meiosis. Mitosis involves one cell division to produce 2 identical diploid cells. Meiosis involves 2 cell divisions producing 4 genetically unique gametes.